So I want to welcome everybody again to the Earth to class for this month. And we're talking about harmful algal blooms or HABs and climate change in the Arctic. Those of you who are not familiar with them, HABs are rapid excessive growths of microscopic algae. They can occur in lakes and oceans throughout the world, warm water, cold water. Many of these organisms contain toxins which can be harmful to aquatic life and through food chains to humans. They've also been called red tides, although many of the microorganisms that cause them are not necessarily red in color. Here's an example of a red tide, which is red. The organisms which cause HABs include cyanobacteria, which are also called blue-green algae, dinoflagellates, and diatoms. Cyanobacteria or blue-green algae are ubiquitous and can be carried in sea spray and travel around the globe. They include the smallest known phytoplankton. Dinoflagellates are organisms with a wide variety, very, very small, as you see from the scales. And diatoms, many of you are familiar with from the fossils that they have left, which have been used for a lot of paleoclimate studies. But the living ones are excellent indicators of water quality, and they produce about 20 to 30% of the oxygen you're breathing right now. There's about 20,000 to 2 million species of diatoms. There's also some other HAB organisms, and I'm not quite certain what these are. If you're exposed to HABs, you can get skin, eye, nose, or throat irritations and neurological symptoms like muscle weakness and dizziness. They can also harm the liver and kidney. If you suspect you have symptoms caused by harmful algal blooms and the toxins, it's wise to contact your healthcare provider or poison control center. HABs cannot be stopped, but in recent years, they've been trying to find various control methods. One involves the use of clays. The clays can get attached to the cells of the microorganisms. They cause them to sink to the sea floor, where they're eventually buried. Another approach involves using nano air bubbles. And in this case, the bubbles, which are smaller than the width of a single human hair, aerate water bodies and create ozone. It helps dissolve harmful algal. Another approach involves a chemical control agent, which is one of the reasons why I shouldn't be editing this late at night. And this chemical agent isolates the mechanisms for the growth, has no impact on other organisms. Location that we're going to study today is one that we've seen before in Earth to Class programs with Chris Zappa. It's in northwestern Alaska in Kotzebue Sound, another name for which is Small Island in English. And I'm not going to try to pronounce the native language. Kotzebue has a population as of 2020 of about 3,000. And there are 3 rivers which flow into Kotzebue Sound. It was originally established as a site for fur trading and other trading among the tribes in the region. But it's part of the US, and if you want to send a letter there, the zip code is 99752. One of the reasons that there's a problem is that many of the people up there still depend upon traditional hunting of seals and other marine organisms and their subsistence hunters. So with climate change, the hunting season has been shortened, and the location where the seals can be found is getting farther and farther away. So Columbia has been working with some of the elder councils up there on a variety of projects, and I have a link here to one of the descriptions of their programs. One of the things they've also been using a lot, as Chris Sappa has informed us in previous Earth to Class programs, is a unoccupied aerial vehicle or drone. Now we're going to find out more about the microorganisms and HAB threats with Dr. Subramaniam. And the webpage has a lot more resources for this. So I'll stop my sharing and I will go to mute. And Ajit, you want to start your sharing?
Thank you all for being here. I'm happy to make this presentation. Hopefully it's going to be a lot of discussion rather than my just talking my whole way through. But there is also a good amount of overlap with some of the stuff that you may have heard from Chris's work, because for me, this is in some fashion the foundations of what we are doing in Kotzebue, and therefore really important to sort of talk about it in the context of co-production of knowledge, because that is really what we need to, how we need to approach this. Now, because I'm using these multiple screens, I don't know if you're seeing. Do you see what I see, which is a screen with just the slide?
Right now, we've got two screens showing up. One has 4 people on a boat, and the other has the article about climate adaptation research.
How about now?
We've got the people on the boat.
That's the only thing you see, right?
Yes.
Okay, so I just wasn't sure which screen I was going to share with the Zoom. So that is what I would like to, that's the screen I want to show. So again, I think it is really important for me to begin any one of these presentations by talking about the importance of co-production of knowledge, especially for a topic such as this, where we're talking about harmful algal blooms that actually impact people's way of life, livelihood, and life. I mean, and in this case, it's true of all three. So so where we are is essentially that the Arctic is changing, and I suspect you may have seen lots of figures to that effect. And actually, oh yes, you would have seen a lot of figures to that effect. But just to make this interactive, can I ask anyone who has seen, if any of you have seen that figure of the declining snow ice cover in the Arctic?
Unmute yourself for responses,
anyone?
please.
No? Well, then I think it's important we begin there, because the context that all of this fits under is basically done through that. So this is essentially the change in ice cover in the Arctic since the 80s. And this is basically taken essentially at the lowest time of year. So this this is basically all the dates you're seeing there is European style. So it is taken late August and early September of each year when the ice cover is at its lowest. And you can see how since the 1980s, the amount of ice in the Arctic has decreased quite substantially. Another way of looking at this, is perhaps an easier way to look at this, is to actually look at the total area of ice that has changed over time. And you can see that since there's been a very significant decrease in the total amount of ice cover. You can also see how it changes over season. So the bit that I was showing you is essentially from this time when it is at the lowest, but you can still see that at the lowest, you had a much higher amount of ice in the 1980s than you have now. So why do we care about any of this? Well, ice is, as you can imagine, fundamental to the way of life of the native peoples that live around the Arctic Circle. They depend on it for transportation, and the tragic fact is that lives are lost on every year because you cannot use a snow machine to go over the ice anymore, or all the traditional paths, roads that they use, roads in quotes, air quotes in case since you can't see me, that they use to go from point A to point B. For example, people in Kotzebue basically go across Kotzebue Sound on snow machines during the winter to get to their various winter camps where they go hunting. And these paths have become unsafe. And while traditional knowledge says that you can potentially go from point A to point B in November, because the ice is really thin or hasn't formed at all, you cannot do that anymore. So as you can imagine, transportation is a major problem. The warming of the Arctic, I suspect you might have seen in terms of housing, has caused the thawing of the tundra, where houses are just collapsing. And so the way you live is not safe anymore. Ice is related to the basic food, that the basic diet of the people that live there, who depend on marine mammals like walruses and seals as a part of their daily meal. And these mammals haul out on the ice, which means that one of two things, and if we have time, I'll get to it. One of two things. One is that if there is no ice to haul out, then the life cycle of the mammal is affected. If the amount of ice that is available to haul out is small, then you end up concentrating the mammals on the ice, which can potentially lead to over harvesting of the mammals because they're much easier to get to. So the lack of ice is basically affecting the marine mammal population and the future of subsistence existence on these mammals going forward in a couple of different ways. So food is a problem, shelter is a problem, transportation is a problem. Given how important it is, and this is a personal belief more than something written anywhere, although I'm trying to make sure that we actually get to write this somewhere. It's my personal belief that all research that is done on ice, we need to be able to do this with the communities that live there and share the results back to the communities that live there in a way that is accessible to them. So these are some of the fundamental principles by which I'm approaching my research on harmful algal blooms in the Arctic. Just again, to reemphasize that point, here's an editorial that was published in the journal Science. And I've just highlighted the bit that I think is really important for us to understand, which is that self-determination and decolonization for communities mean that they control the narrative about how climate crises affect them. It is not good enough for someone like me to sit here in an apartment in New York and talk about how difficult it is for them. We need to work together to figure out what's happening and to come to find solutions, if there are any, to share our findings in an equitable fashion. This is not just something that was done as a editorial in Science. This has also become a part of national policy. So the idea that there is indigenous knowledge, which is also known in the past as traditional ecological knowledge, the fact that we work with communities and we ask communities of what they know has to play into how science is done, and in this case, also how federal policy is set. So there is a memorandum from the Office of Science and Technology in the White House, basically laying this out as an official federal policy. For me, when we talk about co-production of knowledge, we are talking about how we are using our modern methods, such as what satellite remote sensing and what we see here. Sorry about that. So co-production of knowledge, basically, to define it, is a process by which we use indigenous people's knowledge systems and science to, as well as modern science, and in this case, tools like satellite remote sensing, to generate new knowledge and understanding of the world that will not be possible from just using any one of those two systems. It basically, one of the most important things is equity in the research relationship. So the fact that I have access to hundreds of thousands of dollars of research funding that is made available to me from the National Science Foundation, and I parachute into a community where they don't have those resources, does not give me more power to decide what gets done. And so having equity in the relationship is a very important part of this. We have to understand that each of us bring to the table different ways of seeing something and respecting each other's ways of understanding and gaining new knowledge. One of the best ways of describing this is put together by Albert Marshall, who calls it two-eyed seeing, which is essentially seeing with one eye the strengths of indigenous knowledges and ways of knowing, and with the other eye seeing the strengths of Western knowledges and ways of knowing, and to using both eyes together for the benefit of all. I really love this description because I think in the end, this is basically what makes us succeed. And lastly, reciprocity for me is a very important aspect of this two-eyed seeing approach, which basically means mutual respect for contributions from different knowledge systems and their relative importance in generating new understanding of the world. So in the last decade or so, the native peoples living around the Arctic have found a voice and have basically produced a series of documents that lay out the principles that they want followed in the co-production of knowledge. So they basically have said that there will be nothing that can be done there without them. In other words, nothing about us without us. You cannot do research about them, about their way of life, about where they live, without engaging with the people that live there. And to recognize indigenous knowledge as its own. So, and in this case, you might actually hear that there are two, not quite conflicting, but two different ways of seeing it. Because on the one hand, there is the sense that indigenous knowledge is good on its own and it stands on its own legs. On the other hand, there is also a desire that indigenous knowledge is validated by modern or Western or academic knowledge in some fashion, I guess is a good way to put it, because it doesn't seem to be recognized by researchers such as myself as being valid on its own. So there's this idea that we need to validate that. And that's a controversial point that is not accepted by all the native peoples, because some people believe that it is valid by itself. Other people think that having it validated by Western science might actually make it stronger. Practicing good governance is essentially, again, respecting everybody at the table, communicating what you're finding and for communicating with the intent. And I'm putting these things up because these are all relevant to the research that we do on harmful algal blooms. And I hopefully will be able to make that point when we get to the meat of that topic. And building trust. Building trust basically means being able to work with people over time and showing up when you're supposed to, not running away, not parachuting in, doing what you're doing and disappearing. And that basically leads to meaningful partnerships. And these partnerships include sharing of all information, the data, and who owns the information and whose permission you get. And in this particular case, we are talking about pictures taken, photographs taken, and how we use them. And again, I will get to that when I show one other slide coming up. And with the equity in the representation of the Inuit knowledge. Way to think, I mean, so why do we care about any of this? How does this affect, assuming you're working in Antarctica, for example, where there are no people, what is the difference between working in a place like Antarctica where there are no people and therefore there is little what you would think of as traditional or indigenous knowledge versus working in the Arctic, which has been inhabited by people for tens of thousands of years, if not hundreds of thousands of years. And the way to think about that is to use the lens of spatial and temporal scales. I don't know if that makes sense to people what I mean. And so I would really appreciate it if you can unmute yourself and ask a question as I go through this, because these are, I think, useful concepts to have in mind. The idea is that indigenous knowledge is place-based, based on where the community lives. And therefore, that's basically why you're seeing this in the top of the triangle, where you're seeing this as smaller spatial scale, but it has got an extremely wide temporal scale. So it is smaller in spatial scale, but it has a very wide temporal scale because you're talking about generations and generations of knowledge that has been gathered through observation. Whereas when you're doing something like what I do, which is using of satellite data, you're seeing a lot of, you're seeing large spatial scales. We can see the entire Arctic with one satellite scene. However, we're seeing it over a very short period of time. The longest the satellite data goes back is only to the 1980s. We don't have data going back further than that. So it is short on a temporal scale, but wide on a spatial scale. And the question is, how do you sort of combine these two? How do you integrate the large spatial scale that we get from conventional science that is short on temporal scale with the long temporal scale, but space-based that comes from indigenous knowledge? And there's someone who needs to mute themselves.
Deirdre, we're getting background noise from you.
Oh, sorry.
Just so that others can hear me. So here's an illustration that was made in a presentation by Carolina Behe, one of the indigenous writers of the indigenous ways of knowing that I presented, the document I presented before, where she shows this as it's a gorgeous illustration of all the different things that go on in an Inuit community, and how they're all sort of tied to each other. The connection between sea ice thickness and where you find walruses, the animals that you find on the sea floor, such as the crabs, and the animals that you find on the sea surface, like whales that go down to the seafloor to feed and then come back up to the surface. All of this makes up how, and the people that live there and how they are one with the community, all of this makes up how the indigenous knowledge is built. However, when we see this through the eyes of modern science, we end up having whale experts studying the whales, benthic people studying the benthic animals, sociologists studying the people. And the point being that the way Western science is done is fairly fractured with deep expertise, but also siloed where the expertise don't talk to each other. For me, the beauty of this is to understand that the indigenous way of seeing is where it is integrative. It's a systems way of thinking. In engineering, systems engineering is a special field by itself. Indigenous ways of seeing the environment is integrating all that we see in the environment. And for me, this is a very powerful way of understanding the difference between indigenous knowledge and Western knowledge, and how we can talk to each other. Recognizing this is a really key part of that. So having said all that, when we work with our partnerships, we almost always have an advisory council of sorts of knowledge holders who basically we can talk to understand how they see what they see and how we can integrate what they see with what we are observing in our various systems. And this is a story that you may have heard in one of Chris's presentations, but it is again really relevant to what I'm talking about here. And Rosh, so the story that Ross is telling about is that there was a commission to study beaver, to study belugas, and while they were talking about belugas, somebody from the community started talking about beavers, and this panel of outside experts got very confused and told this person, no, stop. We're not talking about beavers, we're talking about belugas. And then this person was able to sort of told, yes, you're talking about belugas, but you can't understand belugas without understanding beavers. And then sort of made the point that the beavers that weren't there were introduced and then became basically an introduced species that took over, and they dammed all the lakes, which basically then prevented the movement of fish. And once the fish basically were not available, then the belugas left. So to understand belugas, you need to understand beavers. And for me, this is a really powerful story again of how indigenous knowledge is able to connect through all of those dots, is able to provide new information on actually what's happening with the whales, whereas the Western way of knowing is really going after what's going on with the whales. And I bring this up again because in the end, what I'm studying is microalgae, which you cannot see with your eye. So microbes are not really a part of the narrative or what people in Kotzebue, for example, talk about. However, they know what it means. They know that under certain conditions, you have a better harvest of fish. So just again, to get us all on the same page, I think this has probably been mentioned before. When I talk about microbes, I'm talking about phytoplankton. And again, to get everybody on the same page, phytoplankton form the base of the food chain. They are the photosynthetic organisms, single cell plants, if you will, that live in the water and are able to use sun's energy through photosynthesis to convert it into organic carbon. And that is then eaten by other organisms. And you basically have the the food web basically developing from that. So changes in phytoplankton community is going to affect the entire food web. And that is one of the issues. If you have, for whatever reason, not enough nutrients available, you have a year when you don't have as much nutrient input, or you have a year that is particularly warm, and so you're not allowing the nutrients to mix up to the surface, then you end up with conditions where you don't have full growth of phytoplankton, which means then you don't have the zooplankton that eat the phytoplankton, you don't have the fish that can eat the zooplankton, so on and so forth. So in the indigenous communities, they have come to associate certain weather patterns with success in hunting, even though the dots that are connected, that connect it all, are not as particularly clear for anybody. What is happening now? So that's the background there. Why do we care about all of this? Last year, there was a cruise that happened last summer where they were looking for toxic phytoplankton, Alexandrium, which actually kills animals and could kill people. So when we say toxic, we are talking about really dangerous. And this is the first time they're seeing as high counts of Alexandrium as you can get in these waters. So to orient you, the colored dots are basically along the ship track, and this is the Chukchi Sea, and Kotzebue is this little. This is the indicating the concentration of cell counts of Alexandrium. And what you see here is right at the mouth to Kotzebue Sound in the Chukchi Sea is some of the places with the highest concentration of Alexandrium. The hypothesis that is formed, that people are using to explain this right now, and this is very recent research. Like I said, this is a cruise that was done last year. This is the first time actual counts have been done in these waters. The hypothesis is that as the water warms, it is bringing these species of harmful algal blooms through the Bering Straits, and this entire area now is seeded with cysts of Alexandrium. And every time you have mixing and the right atmospheric conditions and water conditions, you're able to bring up these cysts that then grow at the surface and become active cells of Alexandrium, which are then eaten by these on the seafloor. They're filter feeders, so they're eaten by clams, which when harvested become a danger to the community. But they're also dangerous because they can be ingested by marine mammals. The good news is that they don't actually accumulate in the fatty tissues, and so it is not as dangerous as it could be. But on the other hand, this has become an issue of major concern. The problem is that this research was done, but it was not done in the communities were not involved with the research from the beginning. The research was initiated, and when they found these things, then the results were sent to the community, or the results were broadcast actually. So they were actually put out in the press. And then there was a little bit of a panic in the community as to what this all means and how to deal with it, what one does with it, with this kind of information. And this continues to be the case. So Western science can go out there and make these measurements, but then how do you sort of give this knowledge back to the community in a way that is actionable? And that is a hanging question right now. This is just an image to show that although the ship track seems to, were just dots along the way, the slightly, I don't know how it shows up on your screens, but the slightly reddish color that you can see in this image shows that that particular bloom could be far more widespread than just along the ship track itself. Now, my work with the community is very much based out of Kotzebue, and I'm working on a slightly different organism that has started being seen in these waters over the last 10 years. These are cyanobacteria, and this is an image that basically shows an example of a bloom screens, but you can see these accumulations that are slightly brighter in color compared to the background. These are surface accumulations of cyanobacteria. And I've been working with a student to develop algorithms. So on the right side, you can see the same image, but that is processed using a satellite algorithm where we can actually pick out. So the bright spots that you see here, the streaks that are whiter that you see are basically areas where we are able to pick out the cyanobacteria using satellite remote sensing algorithms. So on the left-hand side, you're seeing what we call a true color image. So this is essentially what it would look like if you looked out of an airplane window or something like that. And on the right side is the product of an algorithm that we apply to the satellite data to actually extract out the very specific areas where the cyanobacteria blooms are. And for us, of course, the challenge is if it gets cloudy, then there are some difficulties in distinguishing between cyanobacteria and surface blooms. But this is basically the research that we're doing right now as to how to successfully map out cyanobacteria under all different conditions. And to do this, what I've done is worked with community partners there to establish a phytoplankton observatory for these waters. And what I mean by a community-based phytoplankton observatory is that these kinds of cyanobacteria have never been seen in these waters by traditional knowledge, by indigenous knowledge, prior to about 2008 or 2009. But since then, members of the community and the environmental director, Alex Whiting, with whom I work, have noted, he maintains a journal, and he has noted in his journal the annual regular occurrence of cyanobacteria blooms in late August timeframe, just about every year. He basically goes to the pier that is near his house and looks at the water and writes down whatever he sees almost on a daily basis. And he has noticed that we have these blooms happening there in late August or so. What we're doing now is working with Alex and members of the community to go and take water samples from 5 different locations around Kotzebue on a fortnightly basis. This is coming from the fact that the same kind of cyanobacteria have been blooming in the Baltic Sea for thousands and thousands of years. We know thousands and thousands of years because they've actually gotten seafloor cores that basically show that cyanobacteria blooms have been having there for a long time. Baltic is quite a bit south in latitude compared to Kotzebue Sound. So it is possible that we are seeing this transition northward of species that we have not seen before in these waters. And in the Baltic, the research has shown that the intensity of the cyanobacteria blooms depends on all the organisms that occurred before. Again, to sort of walk my way through that, the point is that you have nutrients that come available in the springtime, and you'll have what we call good phytoplankton. I don't want to get all judgy and call something good and something bad, but diatoms do make better food than cyanobacteria. We have blooms of diatoms that happen in the springtime, spring bloom, and they deplete the nutrients. And then in the Baltic, they found that the cyanobacteria come in the fall, and how much cyanobacteria come in depends on the intensity of what happened in terms of the phytoplankton communities before them. So following that logic, we decided that we will actually try and sample for for all the phytoplankton through the entire ice-free season, so that we'll be able to actually understand the causes for the cyanobacteria blooms and try and see if we can work out factors that will actually predict the intensity of these blooms. Why do we care? Cyanobacteria, like I said, I call it bad. It's bad because it's toxic. But even if it were not toxic, and it's not always toxic, even if it was not toxic, it doesn't have the same nutritional content as a diatom. So if your entire diatom population was replaced by cyanobacteria, then the food web basically will have a different structure and would not result in the transfer of energy from photosynthesis through organic carbon, lipids, and fats through to fish in a way that is good for a community. So that's basically why you have this good and bad designations. In this case, we are trying to understand, is the food web going to alter as the phytoplankton community changes? Taking two steps back, why do we care about the phytoplankton? Why do we think the phytoplankton community is changing? It goes back to nutrients. And again, I do not know how basic I need to get with with phytoplankton and nutrients, but I think a very simple way to think about it is your garden. If you're growing a plant, you need to give it fertilizer of some sort for it to grow well. Phytoplankton need nutrients as well. They need nitrogen and phosphorus. And if you have too much of it, you have a harmful algal bloom. That's basically when you have eutrophication and you have blooms that happen when you just have too much nutrients. But if you also have the wrong mixture of nutrients, you can basically have some phytoplankton species growing better than others because they're adapted to that particular mixture of nutrients. In this particular case, we are talking about release of nutrients from the thawing of the tundra. As the tundra thaws, it's releasing the organic material it has in it, which includes organic nitrogen and organic phosphorus. And phytoplankton communities that can use those organic nutrients will thrive in a time when the phytoplankton community that cannot use those nutrients will not. So by thawing the tundra, you're actually changing the phytoplankton community composition in the waters in the Arctic. And in this case, we are working with the hypothesis that the thawing of the tundra is what is causing the cyanobacteria blooms to start growing in these waters right here. But that's a hypothesis, and we need to actually understand if that's true or not. And to that effect, we are basically working on making measurements of not just the phytoplankton, but also we are making measurements of nutrients, we're measuring light and all the other factors that will help us understand how the phytoplankton are growing and where they and how they're doing. So to that end, as I said, we are sampling in a bunch of different locations. So in this satellite image, you can see Kotzebue as the settlement that's right here. And there are two sites right there and right there, which are very close to the community. And we are hoping that we'll actually sample these two sites over the entire year, where they will go out on a boat when it's ice-free to sample all of the sites. But when it's completely covered in ice, they will make holes in the ice like they're doing ice fishing, and will be able to sample the water under the ice at these two locations. In addition, they will go out on a fortnightly basis and sample these other 4 sites. And they will collect water samples from which we will measure nutrients, phytoplankton community composition, looking at it through the microscope, and as well as light, to understand what are the factors that basically promote the growth of phytoplankton in these waters. The background you can also see is image of it's a satellite image where again you can see where all the cyanobacteria blooms are accumulating. One thing you might also notice is that these are some really gorgeous patterns, and this essentially is an indication of the surface circulation pattern in these waters. So the complicated gyres that you see here are surface circulation patterns. On this image, you'll also see the outlets of 2 fairly large rivers. The Kobuk River Delta, which is essentially what you're seeing here, is a big river that drains out of the Brooks Range, and the Noatak is the other river that you can see here that is coming out right here. And our plan, and this is based on knowledge that is given to me by my partners in the community, because we have no measurements in these waters. We are just now beginning to do some of these measurements, and therefore we are completely dependent in a good way. I mean, like when I say completely dependent, I don't mean to suggest there's anything unhappy about it. We are completely dependent in a good way to on the knowledge that they have, and these sites were selected based on our ability to now integrate the outflow of the Noatak River along with the outflow of the Kobuk River. And This way we can understand what the freshwater input into Kotzebue Sound looks like and how this is changing over time. Not only that, but we are also able to figure out that these are all the rivers that are draining the tundra, and therefore we also know what the thawing of the tundra is doing in terms of nutrient supplies. What we found, I would say, maybe surprisingly, when we went and did a bunch of measurements in 2021 at these locations, is that, and these are what you see in this slide are counts of different phytoplankton. So Aphanizomenon and Dolichospermum are cyanobacteria. So, and you can see them here in blue and in red. And the other three organisms that are shown here are diatoms. Normally, we would not be sampling as late as end of September because ice starts forming sometime after mid-September. In this particular case, we were actually testing out a mooring that we wanted to deploy the following year. So I went out there for a couple of days. And so we actually put out the mooring on the 23rd of September and recovered it on the 27th. And so we actually have these two last measurements. If we didn't have these two last measurements, if we just stopped in the middle of September as we normally do, we would have just thought that everything just went down. That was it. Like the ice was starting to form, it is starting to get dark, and therefore there's just no more phytoplankton going on after that. But because by accident we went out there and made these measurements, what we was very surprised to see was something like the concentration of Aphanizomenon went up, as did concentrations of Thalassionema, which is a diatom. Now, diatoms are normally seen to be as good phytoplankton, as I mentioned before, but the beautiful, I don't know if you, have any of you seen pictures of diatoms under a microscope, or seen pictures of diatoms on the internet? If you haven't, I would strongly suggest you go Google diatoms after this talk is over. I did not have any to show you, but maybe I should have put some pictures in there. They are absolutely gorgeous. They're really beautiful. But one of the things that makes them so striking is that they actually have shells made of silica. So these, they're called fristules. These silica shells give them absolutely gorgeous shapes. But you'd also notice that some of the fristules are, they're like literally glass shells, but they're pointed, they're sharp. And this whole idea that we had this bloom of Thalassionema is actually pretty curious, because there was a massive fish kill of herring where tens of thousands of herring ended up washing up on the beaches in early October, along with ice. I mean, like ice had started forming. It was unsafe to go out on a boat, so nobody could go out to take water samples, but there were all these dead fish washing up on the beach. And the question is, what might have killed the fish? Herring is a very important staple food for both the humans as well as they use that for feeding animals. And in this case, Alex had to deal with the question as to whether it was safe to eat herring that was washing up on the beach, and whether it was safe to feed it to the sled dogs that they have there. And it was a difficult one to answer because we do not know if the herring were dying because they were toxic due to the presence of the cyanobacteria Aphanizomenon, or if they were dying because there was a bloom of Thalassionema, which is a problem because what they found when they did autopsy or necropsy, I guess, on the herring was the gills were damaged. And the damage from the gills seemed to come from mechanical abrasion. So one of the questions was whether this was happening because there was this bloom of diatoms that the fish went through and that somehow cut the gills in a way that caused them to die. Late blooms of phytoplankton, again, has never been recorded here, but we don't have much to go by. So to say it was never recorded is a hard one because we just don't have much of a record to go by. That's a low bar. But we found these kinds of organisms in a couple of different places. And this is essentially what our phytoplankton observatory has helped us sort of get going with this kind of new knowledge, where we can combine the measurements we are making with what the community is experiencing, and hopefully we'll be able to provide the information back to the community when questions such as, is it safe to eat, comes up next time. And the other thing that we did was last year we deployed a mooring. By mooring, I mean that you can see this yellow buoy here. It's instrumented with sensors that measures temperature, salinity, phytoplankton chlorophyll concentrations, or phytoplankton concentrations, fluorescence of phycocyanin, which is a pigment found in cyanobacteria. So we'll be able to actually figure out how much cyanobacteria there is, dissolved oxygen, turbidity, a whole bunch of parameters like that. You can also see there that this white thing there is a weather station. And I mentioned that only because we put out the mooring in late June. And so here you can see a plot that shows temperature and salinity at this location where the mooring was, taken every 10 minutes between late June and late September. And you can see that there are these big spikes in temperature along in the mooring, but there is this, there's a peak in the midsummer in mid-June, mid-July, and over time, you can see that it is going down, so it's getting colder, obvious. You can also, what is far more interesting for me is that you can also see these major spikes in salinity. The axis here, you can see this secondary y-axis here. So most of the time, the salinity is below 5, as you can see here. But then there are times when you have these big spikes in salinity that is going over almost 25 parts per thousand. 25 parts per thousand is really salty, especially in the location where this mooring is, which is where you have the outflow of 2 rather large rivers coming, which would make you think that it should be all fresh water. And so these kinds of measurements, like I said, have never been made before. So these are the first time we have continuous records of something like this. But what is, for us, really interesting is the fact that these spikes in salinity that we see there correlate really well with the spikes we see in west winds. So winds blowing out of the west basically are pushing salt water into Kotzebue Sound, and that's basically what is showing up as these big spikes. The one last thing I want to say is this really big spike right here was one that had me giving me heart attacks, because that's when Typhoon Marbek went over this region. I do not know if you had seen pictures in the papers about this typhoon that went over Alaska. Unusual, very unusual to have a typhoon out there. Typhoon is a hurricane in the Pacific. And where there was actually a very striking picture of a house that got lifted out of its foundation and floated down the river and got stuck on a bridge in Nome, which is a community just south of Kotzebue. My fear was that my mooring was not designed to withstand those kinds of wind speeds, or for that matter, the 10-foot storm surge that they predicted, and which ended up happening. And indeed, the mooring was clearly had been sucked underwater, and this weather station that I had is not really designed to be an underwater weather station, and it shorted out. So unfortunately for me, I lost all my weather data from this weather station because it shorted out and it basically lost the data. The other plot that you're seeing here is again just showing the spikes in salinity, but we can now see how the cyanobacteria content, which is basically indicated by the concentrations of phycocyanin, seem to happen immediately after one of these seawater spikes, but has been going up over the time of from midsummer through when we picked it up, and then it basically falls off by the time we get to the end of the September timeframe. Again, these are brand new measurements, as in we have never made them before, and we are trying to get to understanding what they all mean. So to introduce you to what cyanobacteria look like, if you look to the side of a boat, this is basically what the water looks like. For the most part, I think the best way to describe it is as though someone had put grass clippings in the water, is essentially what it would look like. But if you looked under the microscope, these are basically colonies of cyanobacteria, where each of this, like that right there, is a cell. So you have tens to hundreds of cells that basically form a chain, and then you have this big mass of cells that make the bloom near the surface. The region where we've been studying is in Northwest Alaska. So basically that's Alaska, and you're seeing Northwest Alaska there. This is the Hotham Peninsula. And so 2 names, Hotham Peninsula. Then you can see also that many of these have native names, and they have names given by other people. Kotzebue is right there. the moorings were basically located here, and this is Kotzebue Sound, and the Chukchi Sea is out there. We basically are studying these because cyanobacteria can produce cyanotoxins. Cyanotoxins, unlike the saxitoxins that you get from the Alexandrium, don't necessarily kill people. The problem with cyanotoxin is it's a slow and silent killer in that it affects your liver function. And so you might die from liver damage without it ever being actually attributed directly to cyanotoxins. But apart from that, it also changes oxygen concentrations, because while it does photosynthesis near the surface, when it settles into the water column, it decays, and that my bacterial activity that basically does the remineralization of the phytoplankton, the bacteria respire and take down oxygen concentrations. So you end up with sometimes hypoxic conditions deeper in the water column, and that is not good for the benthic animals such as clams and shellfish that live there. Cyanobacterial blooms, like I said, can displace phytoplankton that have better food quality. So you're changing the food quality in the food web, and it can end up disrupting the ecosystem altogether if some key component of the food web fails because the food quality is not good enough. I've been working with a student in developing these satellite algorithms, like I said. So we've basically been processing images from a long period of time, going all the way back to 2005, to try and identify cyanobacteria blooms and trying to identify, how big they are and if they're changing over time. And we basically process the satellite images and to produce maps of this kind. And then we are able to quantify the area that is occupied by the cyanobacteria blooms that you're seeing, that is a result of the algorithm that we expose them to. And we get images where here, in this case, you're just showing the total number of images that are available for each year that are cloud-free between August and September. But we can also use those wind speed that we are getting from the nearby airport and look at how many of the scenes that are clear and that have cyanobacteria blooms. And what you will see here is that we are almost always seeing cyanobacteria blooms when the wind speed is low, and we do not see cyanobacteria blooms when the wind speed goes up. And the reason for this is that when winds pick up, they mix up the cyanobacteria, and so they don't show up at the surface as a bloom anymore. It doesn't mean the cyanobacteria go away, but we cannot use the satellite algorithms to detect them anymore. But even with that, we're able to use a model that gives us a sense of our errors, our error type 1 and type 2 errors in trying to predict when we can see the bloom. And we are basically finding that the area occupied by cyanobacteria blooms have essentially been increasing over time. In other words, this is a problem that is getting worse with we think, the thawing of the tundra. And so with this information, we have now gotten, when we have the big cyanobacteria blooms and what they look like on an annual basis. And the last thing is essentially providing this information back to the community. And in this case, we are working with the community. We are telling the community what we are seeing on a regular basis, like before, when the beginning of the season and end of the season, we do radio interviews. There's a radio station there where the program director there invites us. So we talk to the program director about what we are doing, and that's broadcast. Radio is one of the big ways of communication in that community, which is very remote. And so people rely on the radio for news. We also, when talking to people there, they ask us questions that we would not have thought of asking. And so the idea of working with people there is not just because we want to give them information or we are finding something we want to tell them. We are actually find this engagement is really useful for what we do. And the idea is also that we need to share what we find. And so I go to the school there to talk to the students. I'd actually hope desperately that we'll be able to get a couple of the teachers from the Kotzebues where the regional middle school and high school is. So students or children from the whole Northwest Borough area come and live in Kotzebue and go for middle school and high school. There are primary schools distributed through the communities, but for middle school and high school, they come to Kotzebue. And it has been it has been a real pleasure for me to work with the teachers in Kotzebue, and we end up going to the school to talk to the students and tell them what we do. Sometimes it is just a matter of, this is also a way to make a living. Being an oceanographer is, which is telling them stories of what I do for a living and how I do my research. But that's a really important part. And as has happened with the previous Kakwik Sikukun project, Ice Bridges, we will start producing newsletters once we start having some results. We've only been doing testing so far in the last couple of years, but now we're at a point where we can start writing newsletters with our findings that look like this. These kinds of newsletters are delivered to everyone's mailbox, which is one of the primary ways of communicating out there. And so we will be able to produce these newsletters and deliver them and tell stories about what it is that we're finding. One of the projects that I really would like to start up with the schools there, and this is relevant to all of you, is recently, like in the last 3 years, there's been what we think of as an open source imaging microscope. I'm happy to answer questions on that. So right now, I'm just going to say there's something called the Planktoscope, which is something that you can actually build yourself for a couple of $1,000s. Or in this case, I'm supplying it to the community already built. And I would like to essentially engage with the students there as a class project to take weekly water samples and run it through the Planktoscope. The beauty of the Planktoscope is it's an internet-enabled instrument. So all the images that is captured by the instrument is then uploaded via the internet to a central server, where there is an AI-built software that will identify what that organism is, as long as it is known. So I should explain that again. For all the organisms that has been identified in the system, it'll count them and tell you how much it found. But I'm also working with a colleague who will help train the system. So when all these images are uploaded, we can go and classify the pictures ourselves so we can train the system. The idea is that then the students will have a sense of what's in the water. We're calling this project Making the Invisible Visible, because you may not be completely aware that that teaspoon of water that you have in your hands is actually teeming with life. And this is one way of being able for enabling people to see what's in the water by providing this imaging microscope. And at some point, we are hopeful that these kinds of instruments have been distributed around the world, and all of those images are being accumulated in the central server. So at some point, we hope that students will also be able to compare what they're seeing in their backyard, literally, with what other people are seeing anywhere else in the world, because you can go into this central database and actually click on locations and see what kind of organisms live in other places. So this is a project that we're working with the teachers in Kotzebue. We will hopefully earnestly start doing this coming school year, that is starting in the fall of 2023. But I thought I'll mention this to you to get feedback from you based on your classroom experience on how you think something like this might work and what you think of this idea of having students do a weekly exercise to see what they find in the water as a classroom exercise. And with that, I believe I am done. Am I done?
Thank you very much, Ajit.
Yeah, I'm done.
Okay, I got a couple of questions for you. Number one, has there been any of the students who have gone on to the University of Alaska to get degrees in science?
From the community? Yes.
From the community.
Yes. Actually, it's okay. I'm going to parse that sentence very carefully. That question very carefully. I'm assuming you're thinking about students as in native community students. But
yes.
Do remember that community is made up of not just native people by a whole bunch of people. And so yes, students have gone on to University of Alaska, but it's a matter of what they go off to do, right? There are not that many students that at this point we have managed to interest in environmental science. And even if they were interested in, so I've met with a couple of kids there where they were very interested in environmental science, but there is no course offerings in the Chakchi campus for them to take classes. The only The Chakchi campus is far more what we think of as professional training. And what I mean by that is there is training in health sciences, there's training in software programming or something like that, but not in environmental science. And therefore, even if you're interested in environmental science, you end up having to take a course, or you get your degree in health science. But there's clearly an interesting there.
Excellent. The other question I have is, have you done any piston coring?
Not in these waters, not yet. I mean, that is next project kind of a thing. I want to get this phytoplankton observatory off the ground. We have applied to the NSF, National Science Foundation, 3 times so far. And the challenge that I keep running into is the fact that my peers who are reviewing the proposal are always thinking about all of these projects as conventional science projects. And what I mean by that is they basically are assuming that you have a laboratory, clean laboratory, and you can do genomic sequencing and things like that. We are working in a location where there is not a lab. Most of this work is done in someone's kitchen. And even, I mean, it makes me nervous because even when we add preservatives, which are toxic, doing that next to a place where someone is actually going to prepare food makes me very nervous. So we need to sort of build up the capacity there a little bit before we can start doing fancy things. And so for me, the main objective is to get this phytoplankton observatory off the ground, and then we can get to asking very much more scientifically based questions. But it's interesting you asked that, Mike. Is it something that you're interested in, or is that a question someone asked? Because one of the things that we are finding here is that when my student Anandita did the satellite analysis and went back before 2008, it seemed like there were what looks like maybe phytoplankton cyanobacteria blooms happening in Kotzebue Sound. But we have not found them in Kotzebue Sound in the last 10 years. So there is a hint, if it is indeed cyanobacteria, and we do not know that because there's no ground truth. But if it is indeed cyanobacteria, there's a hint that maybe it used to happen in Kotzebue Sound and is now happening in Kobuk Lake. Like they're shifted geographically. And the only way to get after that is actually doing something like a core to see if we can go back in time to see, to find evidence of cyanobacteria in the cores.
Sounds good. Thank you very much. We've had some nice comments so far. It's open for questions. Everybody can unmute and ask questions.
Shall I go? I see the question from Mary. I'm just looking from the top. The question from Mary is, if the animals exposed to toxins, are they safe to eat? That is the problem, which is that with something like Alexandrium, it is possible, and it's not so much in the animals because it's not gathering in the fat. But if you eat something like the liver or the stomach contents, that's a problem. The other thing is that actually, even with belugas, with whales, or with walruses, you have clams that accumulate in the stomach. And if you end up accidentally eating those, then you have a real problem, because clams are filter feeders, which means they concentrate the Alexandrium and they concentrate the toxins. Whereas, and that's when you run into a real problem. And in the Northeast, for example, so because these kinds of toxic blooms happen in the Gulf of Maine, in the Northeast, when they have something like, when they even detect 2 or 3 cells in the water, just 2 or 3 cells, not 20,000 that you're seeing in the graph that I showed, they shut down the entire fisheries. In Alaska, that's really difficult to do because these are not, for the most part, a lot of this is not commercial fisheries. These are people who go to take food back home. So shutting down fisheries is not as simple, but it is something that we worry about. With cyanotoxins, it's slightly different in that it accumulates, but it's again not something that's toxic because you ate it once. It is toxic because you eat it over a period of time. We are finding these cyanobacteria blooms only since about 2010. So it's maybe it is still a little early to understand how it's going to affect the system because it has not been around for long.
We're open to questions from anyone.
I also had asked about how do you measure the tundra runoff? You said there were 2 things that were really changing from the tundra. I forget, it was nitrogen and something else.
Right. Good question, Mary. So we are going to make 3 different measurements. So we are we know that the tundra runoff is rich in organic matter, right? And so we're going to measure dissolved organic nitrogen, dissolved organic phosphorus, as well as the mooring that I showed you, the buoy that we put out. It has an optical sensor that measures fluorescence of dissolved organic matter. And so we intend to use a series of these measurements to give us a sense of how much of the thawing of the tundra is going through these waters. However, thawing of the tundra is not the only source. In other words, even if the tundra was completely frozen, you're going to have dissolved organic nitrogen, dissolved organic phosphorus, dissolved organic carbon coming out of any terrestrial system. So the trick in this is really distinguishing between what's coming out of the terrestrial system that could happen anyways, what is additionally coming from the thawing of the tundra. There are isotopic signatures that can be used for that. In this case, we're not going there. I mean, like, again, sophisticated scientific questions can be asked, but right now I'm just trying to understand how the phytoplankton community is responding to the changes without getting too deep into the causal factors yet.
Okay. Thank you. In the Mississippi, the river causes algae blooms, enormous ones. So are the rivers possibly doing the same thing here?
Yes, but again, the distinction, well, actually, again, great question, as in, I was going to say the distinction is, and then I stopped myself. The net effect is very similar. The sources are where the difference comes from. The Mississippi is causing what it's causing because of agricultural practices upland, right? A lot of what the nutrients that is causing the phytoplankton blooms in the Mississippi Delta and in the northern Gulf of Mexico is coming from the nitrogen and phosphorus that is used in the Midwest for agricultural purposes. Whereas out here, the population density is probably something like 1 person for 1,000 square kilometers or something like that. So we are not worried, or we are not talking about direct anthropogenic activity. It's indirect, and the source of nutrients is coming from something like the thawing of the tundra, rather than from actual active agriculture. But the net effect is the same: phytoplankton blooms.
Further questions? Yes, Mary.
No, I'm just saying thank you. It's a great presentation. It's really interesting stuff. And I think it's a wonderful idea getting the citizens, the students involved in the water data. That's, I guess it takes a long time for this stuff to percolate, but the interest is there. And if you give them some technology, I'm sure they'll, follow through and help your research by being, local hands.
Thanks, Mary. I mean, actually, I'm curious to hear from any of the other teachers who actually are in the classroom and thinking about how busy your classes are already and the capacity for a teacher to take on extra activities. I mean, like, those are for me the logistical questions that I simply don't have any sense for. I have a good idea, but will it actually work? Curious to know about that. But I also want to make a small, well, I don't know if it's a small distinction, but it's like, it's my research, but it's actually, I think of it as our research. I mean, I really am very sensitive to this idea that I'm going to deploy a bunch of kids to go collect data for me. It is really understanding the environment that we all share, and especially the environment that they live in. That, I mean, again, please help me come up with a language which I can use for this, because I'm struggling with that. I'm not someone that's coming them and giving them technology for enabling my research. And I really would hate if that's how it is seen. I would rather not do it at all. I would like this to be something that helps them understand where they live. Does that sound appropriate? Does that sound reasonable? Does anyone have any comments on that?
Definitely good.
Yeah. I think it's good.
Ajit, are you familiar with the Bright Stars program at AGU?
Yep.
Have any of the students considered submitting for Bright Stars?
Well, I mean, we have not started anything yet, Michael.
Well, even that image, was the artist who did the image that you showed us A student or an adult?
Which one? The satellite
One-
images?
Environmental, the ecosystem.
Oh, no,
Right.
that's one of the elders.
Oh, okay.
Yeah, that was beautiful.
Yeah, no, I just love it. I mean, I love it for any number of reasons, including the fact that it really exposes how the science we do is so siloed, and we need to have an integrated sense to understand how it all works.
Well, several conferences are now putting in art and science sessions. So she might want to consider that, especially as they're being done by Zoom.
Yeah.
Excellent presentation. When we have the recording up, I will share it with everybody. And Ajit, you can share it with the people in Kodasibi.
Sure. Thank you. Kotzebue.
Gatsabu. Okay, so thank you very much. I think we're just about at the end for today. Next one is coming up in March. And we're going to be looking at some interesting ideas that one of the other Lamont scientists are working on. So Ajit, thank you.
Thank you all, and wish you a good weekend.
Thank you very
Yes.
much.
Okay, I'm going to close off now.
Thank you.
Okay, everybody's welcome. Bye.
Thanks so much.